Hi, my name is Alexander Maisiuk, and this is a presentation of my master's thesis. The title of my thesis is Beautiful Degradation by PROTAC technology in carfilzomib-treated multiple myeloma cells. Now, at first sight, this may seem very confusing, so I'd like to split it into parts. And I want to start with the disease that I'm working on, which is multiple myeloma. Multiple myeloma is the second most common cancer of the blood, characterized by clonal expansion of plasma cells. Plasma cells are antibody-producing cells in the bone marrow. The excess antibodies are monoclonal in nature. Most recent available statistics online from 2018 show that there were 469 new patients diagnosed with this disease in Norway. Multiple myeloma is still considered incurable as of 2020. The median overall survival rate since diagnosis is 8.5 years, according to international study. The median age of patients at diagnosis is approximately 66 to 70 years. The Black population seems to be more susceptible to the disease, which is yet to be explained. Why? Women have a lower risk of developing multiple myeloma. The disease is incredibly heterogeneous. People have different types of multiple myeloma, different mutations, gene arrangements, or different number of chromosomes. Multiple myeloma can be divided into two groups, non-hyperdiploid and hyperdiploid. Not only is the disease heterogeneous between people, it is heterogeneous within the same patient. Nowadays, there's a lot of discussion about so-called clonal heterogeneity, which means that the person has a lot of clones within him or herself. These clones dominate during different time points, and sometimes one clone overtakes another over time, especially when the treatment starts due to selection pressure. Multiple myeloma evolves from being asymptomatic to symptomatic by acquiring genetic instability through a series of genetic changes. Oncogenic plasma cell clones affect bone marrow where they initially reside. This in turn enhances bone resorption, which is regulated by osteoclasts and osteoblasts. Multiple myeloma progresses from monoclonal gammopathy of undetermined significance to a smoldering multiple myeloma, followed by plasma cell leukemia. Despite many available treatment options to date, new therapies are still necessary. Coming back to the title, what is BRD4? BRD4 stands for bromodomain containing protein 4, and it is part of BET family proteins which affect proliferation and apoptosis. BET inhibitors disrupt BET family proteins binding to acetylated lysine residues, which in turn leads to less gene expression. BRD4 is an epigenetic regulator protein that associates with chromosomes during mitosis and affects many processes in the cell, like DNA damage and others. BRD4 binds directly to c-Myc promoter region through acetylated histone H3 and induces c-Myc expression. In multiple myeloma cells, BRD4 has been shown to be important to maintain Myc levels. BRD4 also binds to IgH enhancers and other super enhancers. MZ1 is a drug that I'm working with in my project, and it is a JQ1, which is a BRD4 inhibitor based PROTAC developed by the University of Dundee that degrades BRD4 preferentially among other BET family proteins. It has been evaluated in many cancers, including colorectal cancer and multiple myeloma. MZ1 involves proteasomes function. BRD4 degradation by MZ1 leads to reduced binding of BRD4 toward c-Myc promoter and the downregulation of c-Myc. It is important to mention RAS in regards to my project. Now, targeting the RAS signaling pathway is a secondary aim in my thesis, but still important. RAS is a small GTPase, meaning that it hydrolyzes GTP, which leads to downstream signaling through RAF, MEC, ERK in this order. RAS GTPases play an essential role in regulated cell proliferation, differentiation, cell migration, angiogenesis, and apoptosis. RAS is considered a major oncogene in many cancers, not only in multiple myeloma. RAS is not fully pharmacologically accessible yet because of lack of deep pockets for binding of small molecule inhibitors. Therefore, the next effector in the pathway, RAF, and the downstream effector, MEC-ERK, can be utilized in an attempt to limit the signaling pathway. By targeting the RAS pathway, I was trying to achieve two goals. Essentially, achieving BRD4 inhibitors synergy, but also limiting RAS itself because it is a major oncogene, as I have mentioned. Both RAS and MEC are considered undruggable targets. Furthermore, RAS has also dual control over MEC via two independent signaling pathways. First, RAS phosphorylates MEC at serine 62 residue via RAF1 MEC-ERK in this order, extending the half-life of MEC. At the same time, RAS signals through PA3K and AKT in this order to block the activity of JSK3, preventing phosphorylation of MEC at threonine 58 residue and preventing its proteolytic degradation. It is important to mention IL-6 for my project because it is considered a major driver in multiple myeloma. Upon IL-6 binding to its receptor, a signal transduction occurs, which leads to STAT3 phosphorylation at tyrosine 705 site. STAT3 then goes to the nucleus and activates gene transcription. Coming back to the title, what is PROTAC technology? Proteolysis targeting chimeras, that's what they stand for, become increasingly popular. The reason is that for targets such as MEC and RAS, there are obstacles, i.e. large protein-protein interactions, no kinase domain, lack of deep pockets for binding of small molecule inhibitors. PROTACs can overcome challenges of small molecule inhibitors. They may be more potent, selective, sensitive than small molecule inhibitors. Furthermore, PROTACs have been shown to work in humans. And generally speaking, PROTACs consist of three parts. First is a ligand for an E3 ligase, a ligand for a target protein of interest, and also a linker that joins both ligands. Now, in my case, the MZ1 PROTAC binds BRD4, which is a protein of interest, but also it binds Van Hippel-Lindau E3 ligase. Now, I have to say, Van Hippel-Lindau E3 ligase is one of the most used ligases in PROTAC development. Other used E3 ligases include cereblon, but also MDM2. So bringing together the E3 ligase and the target protein results in polyubiquitination of the target protein, which is then labeled for degradation by the proteasome. In my case, bifunctional MZ1 molecule binds simultaneously to the BRD4 and E3 ligase Van Hippel-Lindau, bringing both proteins in proximity and inducing ubiquitination by E2 ubiquitin conjugating enzyme. that has been loaded with ubiquitin. The ubiquitinated BRD4 protein is next degraded by the proteasome, releasing MZ1. Coming back to the title, why did I put carfilzomib in the title? Well, proteasome inhibitors have been successful in the treatment of multiple myeloma, and the response rate is very high. Importantly, multiple myeloma cells respond well to proteasomal inhibitors in comparison to other cancer cell types. Even though there are 3 FDA-approved proteasome inhibitors, bortezomib, carfilzomib, ixazomib, currently available, and others are on the way, like aprozomib, marizabib, delanzomib, and there is issue of cross-resistant between all of them. I chose carfilzomib for combination because it is second-generation irreversible compound. Ixazomib is relatively new, and bortezomib is the first-generation proteasome inhibitor. I had several aims in my thesis. That includes PROTACs combination with a proteasome inhibition controversy, targeting c-MYC indirectly via BRD4, additional targeting of c-MYC indirectly again via RAS signaling pathway, and carfilzomib combination. Several methods have been used in this thesis, which are cell culture, cell titer glow viability assay, Western blot, and phosphoproteins array provided by R&D systems. Results. I have used a panel of 8 myeloma cell lines that differ in terms of growth requirements and cytogenetic background. These cell lines were treated with increasing doses of MZ1 for 48 hours, and the relative cell viability was estimated by measuring ATP levels. Since the cell lines vary in sensitivity towards MZ1, the doses had to be adjusted for each cell line. Although only 8 cell lines were tested in my experiment from approximately 66 multiple myeloma cell lines that are available in the research community, the pattern can be noticed towards IL-6 dependency. IL-6 dependent cell lines used in the laboratory are marked red color, and blue color is for the IL-6 independent cell lines. After finding those response curves for the different myeloma cell lines, I wanted to look closer into the possibility of combining the MZ1 PROTAC with the proteasome inhibitor. This is one of my main aims. In theory, this should be counteractive, since the effect of the PROTAC will depend on a functioning proteasome to degrade the target. This has been reported in the literature. However, I have found that it is possible to combine PROTACs with proteasome inhibitors based on the CTG method. The red circle in this figure demonstrates sort of area of speculation which happens, and I think P38 pathway along with the Hitchcock factor 1 plays a role in a cell protection against proteasome inhibitors. And also this area, it goes in line with the published results that say that proteasome inhibition is counteractive. But the published results are time and dose limited, in my opinion, because as you can see, by increasing the dose, the viability in combination decreases. I observed the same results by Western blot method. In this figure, you can see that with combining MZ1 with carfilzomib, you see more pronounced degradation of CMYC than by MZ1 or carfilzomib alone. It has been reported in the literature that BRD4 inhibitor JQ1 has synergy with the MAP kinase inhibitor trametinib in colorectal cancer. So I was curious, and I tried to combine MZ1 PROTAC with a drug called U0126, which is a MEK inhibitor, but also a weak RAF inhibitor. Well, the idea was with this drug to block the RAS signaling pathway. So I basically observed the same results as in a colorectal article, which is synergy when you block BRD4, when you degrade BRD4 by MZ1, but also when you block RAS signaling pathway. And the synergy is observed with the lower doses of MZ1, in this case 50 nanomolar, whereas what you can see in with 100 nanomolar. And also the synergy can be observed by Western blot as well, which is a more pronounced degradation of CMYC upon combination of two drugs. I wanted to understand better why I saw that it's possible to combine MZ1 and carfilzomib. Therefore, I have used phosphoproteins array provided by RNT systems to see how carfilzomib and MZ1 affect signaling pathway in NMBL6 myeloma cells. This figure is an original photo with highlighted phosphoproteins MSK1, 2, Hitchcock protein 27, Wink1. Phosphoproteins MSK1 and Hitchcock protein 27 belong to P38 pathway, activated by carfilzomib alone and in combination. Wink1 is an unexpected phosphorylated protein by carfilzomib alone and in combination. Image intensity has been measured with image to the light software. I have then averaged signals between two data points for each antibody or reference spot, normalized each of the 8 membranes to its average of the reference spots, removed reference spots from the figure, compared 8 membranes between each other. Signal below 5.3 has been removed. Part A, which is this figure, corresponds to an average of signals from each duplicate antibody from membrane parts A. And I have to mention that from part A figure, the most interesting findings for me were P38 upregulation, MSK, which is part of P38 pathway, Hitchcock protein 27, which is also which also belongs to P38 pathway. Part B corresponds to an average of signals from each duplicate antibody from membrane parts B. And from this figure, most interesting to me was the Wink1 upregulation, both by carfilzomib alone and in combination. But also Hitchcock protein 60. Also STAT3 tyrosine 705. Decrease. So I have then proceeded to validation of the most interesting finding, in my opinion. and I have utilized Western blot. And as you can see, P38 pathway activation is seen upon carfilzomib treatment assessed by Western blot. This experiment has been performed one time. NMBL6 cells were used for 8 hours drug incubation. And I want to point out one interesting finding, that among all these proteins that are upregulated, phospho-MSK1 is the most affected by MZ1 treatment compared to all the other proteins. I have then proceeded to validation of phospho-Wnt1 upregulation by carfilzomib, and I also utilized Western blot. And as you can see on this blot, carfilzomib upregulates phospho-Wnt1. But also at the same time, on the same blot, you can see that phospho-Wnt1 is affected greatly by MZ1 treatment. This experiment has been performed one time. Wnt1 stands for with no lysine kinase 1 and is involved in the regulation of ion transport across cell membranes. However, it has been recently discovered in the literature to be involved in the non-traditional functions. And this is where my interest lies for this protein. I have then proceeded to validation of STAT3 tyrosine 705 decreased by MZ1 because, as I have mentioned, this signaling pathway might reflect IL-6 activity. So the plot shows that STAT3 tyrosine 705 signaling is reduced greatly by MZ1 treatment compared to total STAT3. NBL6 cells were used for 8 hours drug incubation. This experiment has been performed 3 times and is representative. So in conclusion, BRD4 inhibitors need to be used in combination because as it has been reported in the literature and what I observe in my own experiments, there is synergy between BRD4 degradation and RAS signaling pathway blockade. I observe MZ1 effect on c-Myc protein levels. It is possible to combine PROTACs, MZ1 in my case, with proteasome inhibitor, carfilzomib in my case, and I have to say that these results, they go in contrast with the published data by other authors. STAT3 tyrosine 705 signaling is important in myeloma because it might reflect IL-6 activity, and this signaling pathway is affected by MZ1. Wnt1 is an unexpected phosphorylated protein in carfilzomib treated cells, and I think this is fact worth further detailed investigation. Heat shock proteins are important protective response element in myeloma cells to carfilzomib. Carfilzomib activates P38 pathway. So I have therefore fulfilled all of my stated aims, which is a PROTAC combination with the proteasome inhibition controversy, targeting c-Myc indirectly via BRD4, additional targeting of c-Myc indirectly again via RAS signaling pathway, and carfilzomib combination. For future perspective, I would like to check the effect of P38 inhibitor SB 203580 on cleaved caspase 3 protein levels and heat shock factor 1 protein levels with carfilzomib combination. Why am I interested in heat shock factor 1? Because in the literature it has been reported that heat shock factor 1 is a master regulator of all heat shock proteins in the cell. Also, I find interesting to knock out or knock down heat shock factor 1 in an effort to potentiate proteasome inhibitors effect. Wink1 is an unexplored area in cancer research, so I find this direction interesting to follow. Also, I have ideas about resistant mechanisms based on the literature to BRD4 inhibitors. As I mentioned in my thesis, if you go to clinicaltrials.gov today, you can see that a lot of clinical trials that involve BRD4 have status recruiting. So this is a very relevant target today. So I think it's important to start investigating potential resistant mechanism. If this drug, if this target will receive possibly FDA approval in the future. Also, all of my experiments, they need to be repeated with different cell lines, not only NMBL6, even though NMBL6 might be most disease representative cell line from approximately 66 cell lines in research community of myeloma disease. But also my experiments need to be repeated with the primary human cells and in vivo. Thank you.